Good morning, everyone. Our topic is all about determination of L-fucose, rare sugar from Sargassum species. Our work objectives to increase the production of low-calorie rare sugar, to promote the use of potential natural source seaweed, to promote cost-effective production strategy, to promote wide-range application in the pharmaceutical industries. Our work abstract, we collected Sargassum species from Mandapam region, and Fucoidan was obtained by subjecting the samples for soxhlet extraction with different solvents to obtain a good yield of Fucoidan. And to confirm the presence of Fucoidan and L-fucose, the samples were subjected for FTIR and biochemical analysis. Our work novelty, existing work, rare sugar has been produced from several sources such as microorganisms, plants, food industry waste, and whey. Our proposed work is to produce rare sugar from seaweeds, which in turn reduces the production cost. About our project source, Sargassum species are found to have high content of Fucoidan, which is a fucose containing sulfated polysaccharide. Also, fucose is the major monosaccharide present in brown seaweeds. Fucose is also found to have other biological properties which includes anti-inflammatory, anticoagulant, antiviral and anti-diabetic effects. Rare sugar definition given by International Society of Rare Sugars. Rare sugars are monosaccharides and are generally obtained from rare oligosaccharides. The starting materials for the production of rare sugars can be common sugars, polyols, and various building blocks, and it can also be a supramolecule structure. This is the algal classification for the two species preferred. And this is our methodology. We first collected seaweeds from Mandapam coast during low tide in Jan month, and we subjected those two samples for 5 days shade drying. And after shade drying, it was subjected to heat treatment in oven and then ground the samples to granules. The extraction was done with a soxhlet extractor with different solvents such as acetic acid, HCl, ethanol at different normalities. Then to confirm the presence of Fucoidan, the samples were taken for FTIR analysis. These are collected extracts at different normalities. And this is the FTIR analysis results for raw sample of Sargassum longifolium. From this results, it is evident that between the range 3000 and 4000, we confirm the presence of OH groups, and between the range 1000 and 2000, we confirm the presence of ester groups, which in turn confirms the presence of Fucoidan, which is a fucose containing sulfated polysaccharides. And this is for the raw sample of Sargassum YT. Again, it confirms the presence of OH groups and ester groups of Fucoidan. And this is for solid mass after ethanol treatment of Sargassum longifolium, which again gave the results for Fucoidan. And this is for the solid mass of Sargassum longifolium, which confirms the presence of Fucoidan between the required ranges. And this is for the solvent extract of Sargassum longifolium between the ranges 3000 and 2000 OH groups, and between the range 1000 and 2000 again the presence of ester groups in it. This is for the solvent extract of Sargassum YT. And finally, this is the cumulative results for liquid extracts of Sargassum longifolium and YT. Apart from the presence of OH groups and ester groups, we could see the presence of other groups even. This is for the FTIR analysis results of raw sample of Sargassum longifolium and YT, which confirms the presence of groups of Fucoidan. And at last, we did confirmatory biochemical test. In the Molish test, we had red cum violet ring formation at the junction of two layers. This confirms the presence of carbohydrates. And for the Benedict's test, we had green color formation. This confirms the presence of highly concentrated reducing sugar in it. And in the Barford test, we had red precipitate formed at the bottom of the test tube, which confirmed the presence of monosaccharide in it. And for the Anthrone test, again a green color formation, which confirmed the presence of sugar in the sample. And at last, confirmatory test for L-fucose was done, phenol sulfuric acid method. This confirmed the presence of L-fucose by giving a reddish yellow color formation The samples were also read at 480 nanometer in UV, and the absorbance recorded were 1.79 for Sargassum longifolium and 1.75 for Sargassum YT. And these are some of our journal references. Queries. Thank you.